a larger one. All this thing could be maybe enzyme mimics of small molecule, making antibody mimics using AI tools or making shorter versions. So there can be a whole lot of application that come out of it. And I'm not a protein this thing, so I can't do it. But I am looking forward to the group to come out with a plan by which we can actually go forward and do it. And we at Ashoka will be very happy to do anything to help this group function. Of course, Ashoka University will participate. Our AI team, Partha is here, and Subhashis will probably join in. They will all help in setting up in the technology and other aspects of this thing. With this, I'm looking forward to the discussion that we're going to come out with. And again, welcome all of you for this thing. So I'll sort of invite Richi to tell us what is it that Department of Biotechnology thinking about, and what do you?
Yeah, so thank you, sir. Thank you so much, and good afternoon to all the panelists. It's an irony, that in front of all the structural biologists and the pundits of structural biologists, non-structural biologists are speaking in front. That's the irony of Indian bureaucracy, as actually. But what I've said, like to state here is that there are two things. What I'll actually like to mention over here. One is about the BTS network, which of course, Ashoka University has been part of, and most of the members present here have been part of. So as you know that kind of these AI has availed human race with the unprecedented new potentials to solve many global problems and contribute equally to the national goals as well as sustainable development goals. This new network, since most of the members were also the part of the selection process as well, this new network was actually planned. There was already a capacity built up. Till the network was being supported for 30 years, and it was decided that there is already a capacity which is already available, and now we need to dwell on that capacity to actually take it further for R&D purposes and for attaining the larger goals. And with that view, we tried to establish national network projects, which actually, in some cases, I won't say they have not been very successful, but they have been very restricted to the domains of the institute, and institute and all the centers have not made that amount of effort to reach out to their domain vicinity or even beyond those institutes to actually gather the data or probably have those associations which could be translated into meaningful algorithms or probably meaningful outcomes which was actually envisaged. More than that, I would like to state that this has been a very recent discussion actually in the directorate, informatics system directorate, where we are going to hold a mid-review meeting in June or July 2024, wherein, and actually I have just to apprise all the members, I requested Professor Alok for this first seat because I have a meeting with the Secretary DBT at 3 o'clock where probably this plan will be discussed. So wherein, so I was just informing that we are going to have a rigorous mid-review meeting of the centers actually in July or August 2024, wherein we will be, of course, we have the service to the society has been the component for 30 years and that will be there. But the main, a parameter for assessment of these continuation plan will be beyond 2026, of course, for most of the centers. So the continuation plan would be assessed on how the centers have actually made an effort to network itself with the other centers as well as the partnering institutes. And even beyond the NNP project, which has been given to them, has a center made an effort to actually partner with the other institute to gather the data and translate into a meaningful, outcome. So this is one activity which is going to occur in 2020, like mid 2024, and this is what actually I wanted to inform you. And the continuity of the centers, probably, there could be a chance of dropping few centers who are not attaining this target, or probably continuing with, or probably rewarding some other centers who are actually, booming up with this activity. So this could be one information. Second, sir, I would like to, just build upon what you said right now, that, probably all these data which is being generated can be translated into these algorithms, or probably the applications for, use for use by the researchers. So as we need to have the translational part more, because right now department already is in the middle of a new scheme, which is biomanufacturing and biofoundry. And this scheme has been already approved by Government of India. This was announced in the budget speech actually this year, the interim budget speech. And actually there has been a budget of 1,500 crores for 2 years, which has been given to the, actually it was for 3 years, but we could not utilize it this year. So it's 1,500 crores for 2 years, for next 2 years, which has been given. So probably I would request to come up with the translational part more of it. I mean, and also the validation studies, the experimental validation studies, which can actually pertain to the enhancement of biomanufacturing activities. Because of course, all these enzymes and proteins and these structures are going to come up as proteins only, and they are going to be part of the experimental data production only. So I would just request that probably align the activities of the research should be aligned with the scheme, so that we are able to gain maximum out of it. And third, of course, one part which always Dr. Ninave has a message for everybody, that it's duty of each of the center, or probably it's mandate of each of the center, to actually enrich your data as well as your neighboring data into Indian Biological Data Center. And already there is an ISDC, Indian Structural Database, which is already in place. And Dr. Mohanty and Dr. Deepak Nair, they've already made in much of the efforts actually to put in the submission portal in place. And now we are chalking out the sharing guidelines, though not much is there in the data structure guidelines, but still, the framework is already there. So probably that also has to be taken care of, and that would also be the part of assessment criteria when we are doing it in mid 2024. So all these three things is what I wanted to stress upon, and not much to say right now. Thank you.
Thank you. So I'm going to ask expert to make their comments and their suggestion. Essentially on, they can speak on whatever they think is important, but I think I want them to touch on 3 areas. Where do we stand today? How do we increase manpower, trained manpower in this area? What should be the best way to go forward? And what kind of future activities I think we can take it, so that we can demonstrate our capability in this area. So I think the current status, the training of manpower in this area, and then ability to actually demonstrate our capability in a short period of time. If people can touch on that. I'll start with Debasisa. Debasisa, are you there?
Yeah, I'm there. Just a minute. Yes, so I'll just share my screen. Okay, can you see my screen?
Yeah.
Okay, so I mean, since probably we are talking in terms of, how to get a project, projects funded in this area and all this. So, and since Richi already touched upon bio AI and things like that, and he said under biomanufacturing there is scope for I think DVT is thinking of taking focused projects in this area. So I had attended one such discussion meeting. There were a couple of discussion meetings. I think even Anurag attended one probably online. Then in one we visited IIT Guwahati and DVT had a discussion meeting. So the presentation I had put there, I would just put here to run through which are the areas where we can think there is scope for similar protein design or structure-based work. So coming to this one, like what the Richi pointed out, so under biomanufacturing, what DVT is looking for in many of the processes, how AI applications can be enhanced, and some of the areas where we thought one could use any protein design or machine learning or any other computational techniques. These could be the areas like microbial production of fine chemicals and microbiome. So this would require you to analyze biosynthetic pathways and things like that, where this type of AI which we are talking would be useful. And design of nutrition, smart proteins, diet and all, it is one of the big focus area under biomanufacturing. And design of, and that is what protein design, what we are talking of is very much fits there, but one has to link it with the right type of data, experimental data, what sort of experimental data is available, and how do you test the properties of these design proteins. And this is a design of biotherapeutics, especially antibiotics, and I'm not touching drug and vaccines, small molecules and vaccines. People know AI applications has been there, but when it comes to antibody design, and there is a lot of scopes and ABLANG and a lot of language models have been used in the very recently, and people have been able to do like even herceptin binding antibodies. They have been able to design mutations. This antibody binds with higher affinity. That is another area. And these were AMR and the plant science and other things. These are the areas where one this thing. And this is, I have just taken Dr. Vokle's slide, which shows that under biomanufacturing, these are the areas for which I already taught this. I would skip this part. And what seems to be very important for this is, if you go to biomanufacturing, it globally, this has been, people have proposed, it has to go as a design, build, test, learn, and in the cycle, which is not very different from the way we do experiment. We may be doing the individual experiment. We analyze data, design the experiment, then see what went wrong or what is this, go back. So in this area, especially this design and learn, like design, build, and this will probably be in the experimental lab, this will be in biofoundry or something. And but again, this learn and design, these two parts, where bio AI and all this AI protein design we are talking of is going to play a major role. And that is where we can try to focus what we are talking in terms of protein design or protein function prediction or pathway prediction or metabolite prediction into this framework, so that it will be easier for DBT to fit it into one of their thematic areas where they are planning to focus. And this I will skip. And I was just trying to show you some of the interesting things which people have done very recently. Like this is a very simple pathway which is present in chalcone synthase, which makes a molecule like coumarin, but the final product using these 2-3 enzymes, what you get is a interesting fine chemicals, which is of interest to the flavor industry. So you need to deal with 4 enzymes to produce these chemicals in high value. And so now, and if you want to optimize these, like it is not only designing the amino acid sequences of the protein, your strain has to be optimized, your construct has to be optimized, your promoter sequences have to be optimized. What people have been able to do, this paper, what they have shown is they could use a completely automated design strategies, and they could test 65 variants out of 23,823 possible metabolic design combinations, and they could produce this flavonoid with the highest titer as of now seen, 88 mg per liter, which probably in just E. coli, using E. coli, they could produce it. This shows some of the examples how combining a machine learning, protein design, or biosynthetic pathway design, how it is helping. And of course, this is a relatively easy case, you can say. but so there is a lot of scopes in this area. And the other area is, this is something was a bioarchive paper I had noticed, like you have self-driving cars, and this is talking about a self-driving lab for synthetic biology. So this company, it is a startup company. I think it has designed a robotic setup, and it is called Lila. It attempted to build strains that could produce 454 biochemically diverse molecules with precursors located broadly throughout the metabolism of two microbial hosts, Saccharomyces and E. coli. So using this, we can do completely the entire design and testing cycles can be controlled through automation and robotics. I mean, these are probably the dream these things. But what it says is this AI applications has a lot of scopes in these areas. And antibody is one of the app science antibody design, which I already talked, is a area of very active interest to the pharma industry. And I think app science is a company which is entirely based on AI-based design and robotics-based testing of high affinity antibodies. And coming to something like closer to home, what we can do is, of course, a lot of these designs are based on structure and AlphaFold we all talked about, and the power of AlphaFold was known. But subsequently, what ESM fold has come, and which is probably, I mean, AlphaFold was really compute intensive. It required GPUs and things like that. With ESM fold, using language model, you are able to achieve something similar with at a much lesser computational cost. So this can really make these tools available across the desktop, you can say, and type of thing. And what we were telling is like what the type of thing what people have tried developing is large language models based on these sequences, and this is the Progen, which is again a language model based on using the protein sequence space and using UniProt and things like that. And what was specifically interest to me and some of my colleagues like Dr. Devendra Saigal, his lab has been interested in antibody design. And they, of course, have been using traditional bioinformatics methods like structure, which mutations you design. But some of these papers, which are showing recently, if you use a language model-based design, these types of things you are able to design as a constructs, which are giving much more efficient and this one. And what it is telling, like I have looked at some of these methods in little more detail, like the why it is more successful than biophysics-based design. When you go with a biophysics-based design, you calculate it's based on sequence, interaction, finding energy or something. You maybe design a sequence which is not occurring in nature. But by using this language model, any sequence which enters in your design is occurring in nature. And as you know, theoretically, if you generate sequences of a 20 more, it will be 20 to the power 20 possibilities, but all of them do not occur in nature. So the ones which are evolutionarily selected and probably functionally more relevant, they occur. And these types of language models are able to quickly capture that. So that is why this, when it comes to protein design, in addition to structure, these language models have a lot of potential. And this is something which I am interested, but it's little off from this. But some of you could be interested in Ashoka. I'm sure Anurag is interested in. This is the thing called digital twin, which many people have started talking about. But of course, it's a real digital twin. Means okay, you have a mathematical model, and using data and computer model, you are able to simulate the actual biological process as close to the reality as possible. But in biology, this was always a challenge. But with use of data and machine learning, some of the gaps has been filled. So what people have been talking is, people are talking of digital twin of liver, and there are companies which are interested. There is a living heart. There is a digital twin of a heart, which is a Dassault is working. Nokia is working a digital twin of immune system. So these are some of the areas where probably there is potential, but this requires enormous skills in mathematical modeling, which and this is a group like this, we can complement with expertise from people who have expertise in physics, mathematical modeling, machine learning. and people who have data, and that is where we could talk to clinical people or even people who have animal models, immunological data. So that is another area which could be interest, though it is a little off from protein design, which is the talk of our immediate interest. So I think this is there is already a digital immune system project working, and in the Nature NPG Digital Medicine 2022, they already published a roadmap. So they have tried, basically they have tried to incorporate certain number of cell types, certain number of pathogens, and trying to make a mathematical model which would be more predictive than individual models has been there as of today. So, and this is the other types of clinical language models. I would not show it. Anurag gave a very nice talk on using this thing in the work. And I think with this, but finally coming to all these things, which are whether even if you talk a Pro GPT-2 or any of the this one, all it requires large GPU based system. Like at least minimum is 128 NVIDIA 800s, and all these Pro GPT required this much of computing time to be trained and all this. So we need access to have these matches. But there are other models which are people are publishing. Like once they publish this model, there are little data which is coming. Somebody has been able to improvise on those language models, and one is able to train this, either retrain that model using a smaller computing infrastructure, or one is thinking of a different type of embedding where you can achieve the same predictive power at a lesser computing cost. These are the things we could look into it, and I will stop here. And
Thank you, Debasisa. I think we'll probably, in the end, sort of discuss what the various possibilities are that we can take forward. And I would request Debasisa to actually take the summary part of it, and then I think we'll have an open discussion in terms of how do we go forward. Debasisa, Subhashis, are you there?
Yes, I'm there, Alok.
Yeah, Subhashis.
So
Yeah,
I have,
Subhashis, please.
so I have really nothing to add to it. I'll rather play a listening role than try to understand the problem, because, yeah.
So, question is that can our university be a technology hub? I mean, as you see that most of the people are protein structure, this, that, design people, and they do maybe different kinds of computation work. But
yeah, so
is it
I think
possible
that we,
for us to?
yeah, so we have to do the, the language models, or the AlphaFold kind of models for the AI models for proteins and other structure. The advantage is that we have the computing resources, we have biology, we have computer science. The disadvantage is that right now in computer science, none of us know or understand this area well, right? So there'll be a little bit of a learning curve involved for us to pick up this area sufficiently to be able to. So while several of us do general machine learning in this context is something that we have to learn the nuances of it. And we have to start a program. So yes, so I think that we will be quite eager to go in that direction. Given there are at least 4 or 5 machine learning people in the department now. So a more detailed workshop about how to get started will be a good idea.
Yeah, I think one of the ways could be, I mean, as you rightly said, that the people from the biology part will not have much of a understanding of the details of the machine learning, and people like you who have expertise in machine learning have to understand the data and this one. So maybe if we can pick some of the interesting or the challenging state-of-the-art papers which have been published, put it as a journal club where some of our group members present, and which we attend, and then that enhances our understanding both ways. And that could be a starting point to enhance our depth of understanding across the disciplines. And then or after parallelly we take up certain projects in which we can try to work together to see if we what to what extent we will be successful in delivering.
Yes.
So
So maybe
Debashish, what you're suggesting is that you are suggesting a intense workshop focused on the
Yeah.
technology.
yeah.
So
So I think that where we can discuss, take maybe the AlphaFold paper, maybe take and just discuss both the biology and the computer science aspect of it, right, for a whole day, and make plans about how to get started.
Yeah, that could be a starting point.
yeah, I think that's a good idea to do that. And Subhashis, in terms of the country would require future people also.
Yeah, we'll
I know,
require.
but future, we would need people in future to populate other areas where, I mean, other places where.
Yes, so we are desperately trying to recruit faculty, and as you know that in machine learning is a key area, is a hot area for us. But as you know, we are struggling to recruit faculty. Our faculty applications that we are getting are not very much. So one possibility is we can play the hub and get other institutes like India, the IIT Delhi and Triple IT involved, right? And because they don't have the strength in biology, so we can get the interested people and a larger machine learning community from the other IITs and invite them to the workshop. Right? Anurag has done this before, so he will know. Because this will require machine learning of not just an use of machine learning, but I think that we'll have to get into the machine learning algorithms in fairly detailed. So Anurag, what do you suggest?
No, it's been very interesting, Subhashish, to look at this.
Mm.
And so when I look at the people who did, for example, protein language models, only a few of them were hard CS, many of them weren't, but the team requires all types of people. And sometimes I wonder, in the current generation of technology where nobody actually fully understands what is really happening except that it is happening, maybe a thoughtful curation of what these models can learn from maybe about as good an approach because, for example, now that they have MedLlama as a completely open source version that people are doing 0 shots and few shot learnings on. I mean, the type of stuff that is coming out of it is such, I don't think even CS people can really explain what is coming out of it.
Yeah.
At least
any LLM model, yeah.
So I think one of the strengths that we could bring in, and there are 3 different ways of going about it. And then let me, before starting that, say, because Alok wanted us to start from where we are. So where we are, Alok, is in not the ideal place. Both DBT's biomanufacturing is impeded by the fact that while we have platforms, we haven't really done much of it. ICMR within the next few months will announce first in the world grants. Because almost nothing that is done is first in the world. And they realize that until something is done that is first in the world, India cannot get out of that middle income trap that Dr. Gokhale speaks about. So in fact, they will launch this category called only first in the world grants. The only requirement to apply is that whatever you're proposing should be first in the world. They are expecting 95% of it to fail. Now, with this context, let me come back to Subhashish's point. We probably would not be able to compete in the realm of hiring the genuine CS guys specialized in the area that is now already booming. It simply won't work that way. We can't. On the other hand, given how few places in India can genuinely teach CS to people coming from biological backgrounds but interested, and young people willing to experiment and try who don't know that it can't be done yet, we could do some first in the world things by the enthusiasm of young people who aren't bound by knowledge of the impossible.
Yeah.
So, and I think that's where Ashoka could best position itself by bringing in genuine CS people, few in number, but using them along with a larger biology department with interested people to create even larger number of early career or young people who will play with this. And I honestly think that as people learned in the startup ecosystem, lots of young people playing with things in today's time,
Will
See,
generate something.
as well as any other model in this world.
Yeah, especially in an area like machine learning, which is
Absolutely.
theory-less, right? Yeah. Which is
And especially the current versions, which actually also require
Hmm.
a large community to form on the open source ones, like Alkapa, or because everybody is using GPT.
Hmm.
But everybody is using GPT. GPT is getting enriched. But on the other hand, MedLlama will give you true control over the IP that you generate on a MedLlama, because you can run the model, fine-tune it, multi-shot it on your own computers. So I think if we were to make a push, make people comfortable with actually tuning their own models on their own systems, which is one level beyond GPT, but simple enough that you don't need as much hard CS as is required, and focus on that sweet spot, we could make a difference.
It
through
does not require any hard CS, but it requires
understanding. It requires understanding.
understanding, playing
Yes.
around with the yeah.
And so guided playing with people who have genuine understanding is a good thing that Ashoka could focus on in this intersection, because we have that mix. In fact, Debashish probably would be the first to say that if NII went out to recruit a pure CS person, or IGIB went out to recruit a pure CS person, we would have an even harder time than an Ashoka. So, and reality is that IIT Delhi also has not done extremely well when it comes to biological recruitments. So I think Ashoka will not necessarily get the absolute peak in one specialized area, but as a good mix, we could help create along with other people. So I think that's a long way of saying what Subhashis said. We need to act as a way of enabling the community. And I think that's what Alok has in mind through this.
So I think that getting other CS people involved in the endeavor, right?
Yes, yes.
In a workshop,
Mhm.
suitably planned workshop, the younger people who have worked with LLMs, right? And
Yep.
making a team with the biologists so that they can play around on GPU machines is the way to go.
Yeah. And a wonderful person, Debarka Sen Gupta at IIIT Delhi, has been extremely willing to be part of such efforts. And he is possibly one of the best in the community. I think Debashish might agree in terms of who comes from a statistics CS background into biology. So he's another invaluable resource who is willing to spend time with us on doing this.
I'll just add one small comment. So AlphaFold is mostly closed source. The newer models, AlphaFold Multimer and AlphaFold All Atom. But there are like a bunch of open source projects. So there is a community called CollabFold, led by somebody from Harvard. And the community and the Baker Lab has also put most of their code, David Baker, who leads Institute of Protein Design at University of Washington, which kind of somehow in some way pioneered the whole field of protein design. So all their code is open source. So as Professor Anurag was saying, that the future of generative AI, in fact, most AI is open source models harnessed for a particular application, as Professor Alok was saying, for designing therapeutics, designing AAV vectors for gene therapy. So we have to kind of build that capability of playing around, fine-tuning open source models like Llama, like CollabFold, specifically.
Yes, the whole world is doing that. That's nothing new. In every domain that is happening. So we have to just get that started somehow.
Yeah, right.
And getting that started without CS people will not be easy, right? I mean, so we have to get some of the CS folks. Devarga will be interested.
Start that, Subhashis and Anurag, I think we'll take it forward, start it. And Debashish and others will also, I guess, involved in it. And I think I will ask Shachi to from NCBS to present her thoughts. Shachi?
Hi. Yeah, I don't really have a presentation of any sort. I don't know whether you can see me now. I just here we go.
Can see you, ma'am.
Yeah. So you can hear me, right? Even if you can't see me, maybe you can.
And we can see you as well as you.
Also, okay, very good. Okay, so I have been actually doing some protein design for a while now. And when I say protein design, I usually mean whole protein design, like sort of saying, okay, I don't really care about what the function of the protein is. I just want to get the structure right. And this is sort of very traditional, I mean, consensus design, all of this stuff. In fact, so one of the things that I found very early on is that it's very difficult to have a pipeline which goes from theory to experiment and back. I set up a lab in NCBS to do this with having no sort of experimental background. And this was really hard. So sometimes, I mean, when you have a computational background, picking up other computational methods is fairly easy. But sort of having a close collaboration with somebody who's experimental, who can do the experiments, who's interested in the experiments, and when there is some feedback between these two, this is very important, and it's sort of hard to come by. So this is, I mean, this is sort of my own experience with this. So if you're going to set up a center of some sort, it would be nice to have whatever proteins are being designed, being synthesized and put into the lab and looked at in some way quickly. I think this is very important. And so having the experimental people and not just biologists where you're putting it into, but I mean, if you're thinking in terms of protein design as in structure design, then it's also important to have all the structural biologists who will maybe look at the protein or whatever it is, but who are experimental to do this. That's one thing. In terms of the question of whether we want to have our own program of doing, meaning have our own model of doing protein design, and so on. So build our own sort of neural network. It's something that you might do. It's something that you might not do. It's because you can actually use many of these AlphaFold and Rosetta Fold and sort of build your design pipeline on top of them. So that's, yeah, I mean, as a first step, one can consider that before one goes off and sort of builds one's own language. I mean, whatever model, not necessarily language, but any model that that you want, any deep neural network that you want to do the design. So so that's sort of where I think, I mean, where, as in what my thoughts are about where we are today. In terms of training, training is sort of, I mean, I don't know, I have money. I find sometimes that I have money, but I find it very hard to find people. Is there a question?
Go ahead.
Yeah, okay. So in terms of training, I think that's actually very important. And I don't really have very clear ideas of how to do it, but I think it's very important not only for sort of future, I think it's also important for present, in the sense that it's sort of difficult to find people who will, I mean, at some point, you run out of ideas and you really want other people to pitch in. And I think this whole idea of getting even like interns, 1-year interns from undergraduate programs who are just willing to do whatever it is and play around and sort of roping in the enthusiasm of the young. And they tend to talk to each other much more easily, and they have weird ideas which sometimes are the best ones. So even, I would say, even like an internship program where you bring in people, undergraduates for thesis, but pay them so that they are sort of excited about coming in and doing either the language models or the or the biology synthesizing and purifying proteins, or both is sort of, I would say would be nice to have, so that these people can then come in and see the work that's being done and then feed into maybe either for further work here or elsewhere, as it may be. So the third one, I have the ability to demonstrate capability. So I think this is very different for biologists, and different for the government, as in if you're going to show things about what have you done, then it has to have some relevance to human health, or it has to have some relevance to applications, translations, human health, versus capability, where I am excited about, look, I have a new structure. And a lot of the protein design that's maybe coming out in Science and Nature and so on is at this level right now, which is that, meaning there are a few applications that are out there, but a lot of it is sort of blue sky in some sense. But if you have the capability of doing blue sky, I think you can very quickly ramp up to applications when a very appropriate or whatever, when your blue sky science or whatever fits in quickly to an application. So you're then poised to do some applications when they arise. So I think, meaning I don't have much to say in terms of ability to demonstrate capability, in terms of putting something into the clinic, or doing other things. But the other thoughts I have had about protein design so far is that it's much easier to do things where you're doing them outside the human body. So essentially, if you want to have binders, or if you want to create tests for something where you're adding blood to some, meaning, even if it's human blood. So biomedical devices, things in industry where all of these are much easier applications in terms of any new biological design, then sort of medical applications where you're trying to put a protein into the body and then you don't know. That takes too long. So I would say, if you want to quickly demonstrate capability, then maybe that's the direction you want to look at. So I think that's all I have to say.
Thanks very much. Now, Lipi.
Yeah. Hi, everyone. Yeah, it's very interesting to, be here. Also to hear the thoughts of everyone, including Debasisa, Shachi, and the thoughts by DBT, where they're heading. So I'm just going to remove the component by DBT and how and when that. I'm going to pick from what Alok said, that where are we today in terms of protein design? And that's where I'm going to start. So some of the work, we are using some of the terminologies in a vague manner. For instance, the AF2, protein design, AI, LLMs, they all may be very hype word right now in structural biology because there is a huge increase in data sets. And that is primarily because of AlphaFold 2, given that Google came with DeepMind. Now, some of the LLMs in parallel to AI-based AF2 were developed, which is what Debashish mentioned. We're also targeting generation of protein structures. So let's keep that generation of protein structures as one of the theme. And I think we have in no way we can compete them in terms of generation of this tool set, along with CS folks, because they're really heavy into this. In fact, there are multiple teams at MNCs like Amazon and everyone where they're trying to understand parallelly how can we get better than AF2. There are multiple problems which are not solved there. For example, large proteins are not predicted with accuracy. Transmembrane domains are not given accessibility, and they have no relevance. And there are other issues with intrinsically disordered regions, how domain to domain contacts are occurring. There are some areas where the homology is little. So there is one entire theme of generation of protein structures. Given the impetus by AF2, we have now 48 proteomes. So the data set is in millions. There is a parallel understanding of tool development here. So this, I want to put it in one bucket. We have one publication in this area where we have worked around with AF2 in the area of autophagy. So we've tried to give, so the gist of the publication there is this, that the structures which are coming out are really of high quality in a 50% stage. So you have the whole pathway available to you, and you can now say that 50% of structural models are of high accuracy. That's the data set that we presented. We moved the entire bit till experimental validation, and it's unbelievable that the pace to what you can reach to validation is very fast. You don't have to go for fragment-based homology anymore. So there's large-scale structural data set already available. And that's what we argued for, that now you need to re-navigate your biological questions such that you utilize this data set. So I think that's one bucket which is there. Now this bucket may be linked to protein design, but parallelly protein design is completely a different game altogether, which may or may not be linked to this. So in protein design, it's amazing how much data is not able to be you can say reproducible. Even the best of best paper, you pick it up and you tell me if they have the pipeline that you can reproduce. So largely what Shachi also said, it's a back and forth with experimental validations. A lot of good papers here in the design. So I'm again removing the tool component here. Again, I'm saying we may not be able to compete in the tool domain. So one of my students, she went to Chile where David Baker and other folks had a workshop, and she was there in Chile, and then they gave her a lot of workshop tutorials on RF diffusion. Now she comes back with all the insights about how we can design using these tools. And it's unbelievable that the backbone and the accuracy with which, the folds can now be designed. But there is a lot of scope there. Those folks know the open questions and they're working on it. Side chains are not to accuracy. We do not know where to place them. There are a lot of steric clashes, but I mean, there are open questions, but I'm saying even addressing that is being done by multiple labs. And so here we may not be relevant, but if we have a given question, which Debasisa mentioned from his topics of interest, for example, I'm taking EMR or antibody design, enzyme engineering, which Professor Alok. So in terms of those, there is a lot of India-specific variations one could work on. Multivalent antibodies that are guided by the data generated within India. And that needs back and forth understanding of not just the omics data, but also the understanding of how those variations can now produce the epitope structures or design epitope structures that can produce multivalent antibodies. So antibody design is clearly in the scope of the work which one could do. And there are other areas in which also you mentioned, but enzyme engineering again is very competitive there because the folks have been working with bioorthogonal PROTACs largely in targeted protein degradation space. So there might be a different scientific discussion altogether. But if we just consider de novo design with targeted function, this needs intense biological collaborations. And these collaborations have to be hand in hand. Like really every day you synthesize, and Shachi and I have sometimes discussed about once challenges of doing this back and forth. It really has to be a dedicated experimental lab for validation. And you end up having your own lab. So as computational biologist, I'm also having now a validation person, a crystallographer working with me now, who's designing it. So it's the tables have slightly turned, if I may say so. And you give them the data set. So that's the current state where we are. We have to probably work alongside with the experimental folks. And so this covers the AI and protein design components. Lastly, just second thing which Alok said was that in terms of manpower training, I'll give you an example of my recent few recruits have been purely from CS background and also senior folks like postdoc. So one of my postdoc comes from engineering background, and she comes from the area where the classifying of images was the main topic. So she understood what is what, right? Structures was completely a different thing altogether. So she could cry on the first day of the lab meeting to see structures, what are the structures. So introduction of ML and AI trained folks with structures is really hard. They completely don't understand that language at all. Sometimes genomics is easier because, it's text. But with proteins, even explaining conformational changes, that this could be now a potential topic for distinguishing mutations, for example, it has taken me a year. And now she comes up with transformers and we have a very interesting discussion. But my experience has been 1 year, back and forth every day, sitting with the person, making them understand what is the conformational change, what is the chi angle, because sometimes you have to now come at supervised learning, unsupervised learning. But what do you tell in terms of supervised learning if the person doesn't know anything about side chains, side chain dihedral? So, protein structures are much more complex. And for that reason comes my third point, that the state where we are, and I think we can get started here, that our biology labs are very strong, at least in the Delhi hub. So if we get some of these questions very clear, that what is the biological function that we would like to modulate? Okay, that X function being modulated, where do we want to start with? And I think there are a lot of scope in this direction. We could pick up any of the topics which Dr. Mohanty said. So that's where we are in terms of scope of protein design, AF2, also training. And lastly, you talked about future activities. And so future activities, there are few papers which I could share, but largely also from the Institute of Protein Design, David Baker Lab, they have moved at a pace unimaginable in terms of application space also. So what they are doing in those future activities are they're tuning protein functions with the capacity to sense the protein structures within the human body are able to now sense more and more ligands. So that's very interesting, some of these works that are coming up. So where you are designing protein sensors, and some of their work, we've discussed that in the lab meetings. But yeah, I mean, it's very, very interesting. Rosetta is available on as an open source, but how they intuitively go back and forth with the design and coming back is really challenging. So, they all, not all, everything is online as of now. So future activities to tune protein functions is very interesting for me. And that is a scope with close collaboration with experimental labs, I think is a very interesting prospect. That's where I like to end.
Okay, thanks, Lipi. Dinesh?
Yeah, sure. I think the speakers before me have summarized the issue in front of us very well, and I didn't know the exact purpose of the workshop, but I have read the papers which Ashish shared with me, and I will like to share my experiences with AlphaFold because I work in a field where we model a lot of plasmodium proteins. And some of the proteins are very nasty that their structures are nowhere possible to be modeled. And we used all the possible modeling software, including threading, AlphaFold, RobettaFold, etc. And we met with very little success because of the fact that these proteins are very strange in nature. They are different from their homologs in rest of the genomes which are very close to them. So for example, we worked on a protein called P. falciparum circumsporosite protein, which is around 400 amino acids long, but it is characterized with lot of Na and P repeats. So this protein was very hard to model, and we tried AlphaFold also, where we met with very little success. Most of the regions lied in very low PLDDT of even less than 50, so which indicates that structure was not usable at all. And then we used modeler, which again gave lots of regions which were not lying in Ramachandran plot, or RMSD with the templates were very high, which also indicates poor quality of the modeling. And then we also tried ITSR, which surprisingly gave good results. So my experience with AlphaFold is quite mixed as compared to the initial hype about the structure. Maybe other people who are working on different human proteins, they might have found it very useful. But for my purpose of modeling Plasmodium protein, it has been not a good performer. So I have gone through the papers and found the following challenges, which are true for all the protein folding problem in general, excluding if you forget about modeling the malaria protein. For example, these models lack physical insights into protein folding problem. So the models which are developed using AI are actually black boxes. They don't give insights into how the protein is folding. So this is crux of the papers which Ashish has shared with us. And it is very difficult to model, especially the flexible and dynamic proteins, which I think Debashish and Lipi also emphasized, because these are very flexible proteins. And if you make any small mutations, the AI models are not able to precisely predict the change in conformation. And especially the proteins which are dynamic in nature, for example, viral proteins or antibodies, it is very hard to predict the correct structure based on changing amino acid sequences. So this is mainly because the current methods which are developed using AI are mainly alignment dependent. They are based on multiple sequence alignment of proteins for which structures are known. So they are unable to predict effect of mutations also, because if you make subtle mutations in these aligned proteins, the AI or any other model using machine learning or any other prediction model, they are unable to predict the change in conformation, because they are totally relying on multiple sequence alignments which are based on similarity based methods. So the another challenge is how to take care of these problems. And there is another issue of how to integrate machine learning with physics-based methods, because after all, experiments have been used in the past for making some cues to or improve the homology-based modeling, for example, NMR and so on. So how do you integrate such valuable inferences from experiments to integrate them with machine learning? So this is one of the biggest challenge, and which, if solved, can be a hallmark in improving the efficiency of these methods. So another issue is that can the future methods predict cascade interactions amongst the protein? For example, if you make a small change in a protein, what will be the downstream effect of this change in the protein conformation and its interaction with its downstream interacting partners? So this much about the AI for protein fold. So uh, previous speakers talk about how to make Ashoka better in terms of improving its AI capabilities for biology and protein folding in general. So I would suggest that you organize a workshop for training the trainers. For example, whatever existing manpower you have, it will be better to send them to centers which are already known to be leaders in their field. For example, Anurag mentioned Debasisa Gupta. He's a very good trainer, and perhaps a few faculty from Ashoka can be sent to his lab for a couple of weeks to get trained in the techniques they are expert at. And there could be also joint workshops for longer duration. So because 3 or 4 days are hardly anything. They don't teach much about the intricacies of AI and how to use it into the problems faced by a biologist or the biological data. So I propose that you hold joint workshops of longer duration, and we can also organize hackathons in, say, example, pure IT institutes, triple IT or IIT Delhi and so on, where you can enthuse students to solve problems in biology. Maybe some of them will be more than willing to join Ashoka to pursue their PhD, and they could be very useful human resource for developing such programs. And lastly, I and Debashish are associated with IBDC project, where we have built a large infrastructure for compute power. So if Ashoka is uh, desiring to use this facility, the facility can be made available to Ashoka faculty for running their projects in AI in biology, because we do have a battery of DGX machines which are largely unutilized or underutilized at the moment. We'll be happy to provide access to the machines whenever they are free. So, and commenting on Lipi's experience with hiring pure CS students into her lab, I have the contradictory experience that I have been recruiting only pure biology students, and I have been lucky to get students which are able to learn various computational techniques very nicely, and they have been very productive because they don't have to be taught about various biological concepts, but they are more than willing to use AI algorithms, which are anyway ready-made. You don't have to write or code from beginning. The AI algorithms are already there. You just have to train them with formatted data sets. The formats of these algorithms are very simple. The format in which the input has to be fed into these algorithms is very simple, and the way to evaluate these performance of these algorithms is also very simple. So I think it depends on the interest of the students, whether they are from CS or biology, doesn't matter as far as data science and AI is concerned. So that's all from my side. Thank you.
Thanks, Dinesh. We have Dilip from Ashoka.
Yeah. Hi, can you hear me? So I'm here in an interesting position, I believe, except Alok, I am only the experimental biologist who has been basically purifying and working on the structures of proteins. And I am mostly the end user of all these programs. And my experience with the AlphaFold has been a mixed feeling. So when you have a very nice, well-behaving protein, you predict the structure. If it's 400, 300 amino acid, the structure is as good as high-resolution X-ray structure or cryo-EM structure. The moment you jump into the unknown territory, for viruses, I work on viruses, we work on non-structural proteins. The moment you go into that territory, AlphaFold is as bad as any other tool. And that is where probably it needs to improve, or protein prediction models need to improve. My comment for where are we actually, and if we look into whole world, the only academic group which has been doing exceptionally well in terms of protein designing and prediction in world is David Becker's group. And if you look in his group, there is more than 50 people, and there's more than 1 dozen people. There are more than 1 dozen people who just work on this experimental validation of things. Coming to the lipids point, the back and fro of all the workflow, what they do is when they predict any structure, they have already a team which will be validating those structures. And that is where probably we cannot have a group of 50 people, but we need to come up with some sort of consensus where we have a seamless collaboration between experimental biologists to validate whatever we are finding in terms of our AI-based tools. Now, the question, as Lipi also pointed out, it's more important to find out the question, and it would be good if we have a question which can be easily addressable. At the moment, I believe antibody-based designing is one of those tools. From the computational biology, it would be easier. Just yesterday, there was a preprint from David Baker's group where they have designed a de novo antibody fragment against a novel epitope. And they also did the cryo-EM structure of that design antibody with that particular protein. Now, I believe antibodies could be very well a very nice target for two reasons. First of all, they are easily workable. If we have single chain variable fragments, they are easily expressible in the prokaryotic system. And also they help to stabilize the protein, which may have inherent instability. So that question, I believe, for picking up for all of us as a group to design any sort of therapeutics, I believe that's one open thing that where we should be looking into it. And about how we can train more manpower, I also agree with the Professor Dinesh Gupta that it has to be dependent on the person who's interested, and we don't have to train all computational biologists, computational people to learn biology or biologist people to learn computation. It would be enough for us if we can just train the biologists to use these tools effectively, to tell them what are the questions that you can address by using these tools so that they can design better questions. And if there is an interested computational biologist, we may also involve them to develop these tools better or make them better by using the existing tools. So I believe we need to design some sort of a workshop where we can come at the interface and we can, both of these groups can be benefited. And the in terms of activities, yeah, as all the people suggested, it's better if we have more regular meetings, and if it is possible to have a joint once in a month type of journal club where computational people will be talking about these language models, where biology people can tell what are the different discoveries that is coming using all these tools, and we may basically find a common ground. So I believe most of the topics have already been covered, which I wrote here. So yeah, that is where I'd like to end. And yeah, thank you. Thank you, Ashish, for organizing this.
Thanks. I think we have sort of finished. Anybody wants to make another comment, or before I ask Debasisa to summarize and tell us how to go forward? I mean, I just take up one point that Dinesh also said, Dilip said, and some other people, Lipi, has been talking about. Before I give it to Partha can actually, he's from computer science, so he can tell us a little bit more about it. But I think it may be a good idea in Ashoka if we, and which could be very good, if for example, biology offers courses. Biology is already offering courses for computer science students. Maybe we can offer courses on proteins, etc., which a lot of computer science students can take, and also offer some basic computation course, which we are anyway offering to our biology student. I think there's some amount of cross-discipline to it. But if you want to do in-depth work, you do need people, particularly if you want to develop tools, methods, etc., or the codes. You need computer science people to understand what we are doing and what we're doing. Partha, are you there?
Good afternoon. Thank you very much, Alok. Am I audible?
Yeah.
Yeah. So as Subhashis said, I'm also here more as a learner because this domain has excited me always, but I never looked into it seriously, particularly from the computational perspectives. Learning this, I fully agree with the suggestion of the workshop and going deeper. As a computer science professor here, and working in some domains with AI, and particularly from the Center for Data Science and Analytics, whose main charter is to facilitate the use of different AI, machine learning, and data science tools for different application areas. I would be very interested to work with all of you to design such programs, take part in that, and also facilitate building the infrastructure. But as I said, personally, I'm not at all an expert. I'm just a beginner learner here. So I will initially be just a facilitator, and then as many of the experts have already expressed the opinion that it's very difficult to teach biology to computer scientists, which I agree. And the main reason that we want our students to learn biology. so that they don't get into the similar difficult situation. So please let us know how I can help. But certainly the Data Science Center is taking a lot of initiatives to create capacity building programs in different areas. We have already run programs in social sector. We are continuing on that, but this will be a lot more serious one. And I would be happy to join coordinating this and making this happen. I think otherwise, a lot of experts have told things which I'm yet to digest in reality. Right.
Thank you. We need your help.
Yeah, sure.
Anyway.
Of course. I mean, in Ashoka, there's a great opportunity for all of us being together and being so willing to work together on this, which is a very, very imminent problem. It's a great opportunity for us who talk about AI, machine learning, and all of that, because biology, in my view, in the view of Subhashis, has also always been talking about it, is the great opportunity for computer science and AI to move forward. The real problems lie there. But in terms of protein design and the related issues that we have been talking about, we need to learn how we can actually get involved. So slowly we will start working with some of you and get educated and start contributing to the research that you are already doing. Certainly.
Thanks. Debasisa, it's you summarize it, you tell us what to do after this.
This difficult task. Sorry. Yeah, so thanks to Alok, Ashish, and Anurag for arranging this meeting. I think it has been a good exercise to listen to each other. I think, we, each of us pointed out what are the real difficulties. And of course, we need to have people with computer science expertise, and computer science have to learn biology, biologists have to learn computer science or this thing. But if you think each of these in the ideal world, it will never be possible. No computer scientist will sit down to do a full course in biology or spend enough time to read biology, and same would not hold good other way around. So we need to find this. It is like the on-the-job learning, like the way Lipi gave example, her postdoc from computer science who came, she doesn't understand structure, and whichever structural biology parts she required, she gave the inputs on that, and that is the best way to learn. And as Anurag gave example, none of the guys who have done outstanding work in this area, they would see they are either be full-fledged hardcore trained computer scientist or this. Even if you go to AlphaFold, John Jumper is the guy who had spent his career on biophysics-based protein folding. He decided to leave this field and go and work in financial modeling and other areas, ended up in Google. And when Google started this DeepMind and protein structure prediction project, so he was the right man who had the understanding of protein structure. He came and led the project. So the way the field develops like this. So we need to, interaction is the only way we can develop expertise. And also that is the point we need to start. And the other point is, of course, AlphaFold has not solved all the problem. It has its limitations and all that. But we have to take a global picture. Like a lot of these people like us who have worked on drug design, and coming up with a new drug based on a computational prediction is very difficult. And even if AI, it is equally difficult. But now if you see, each and every company is spending huge investment on AI-based drug design. So we need to, I'm sure there is some amount of hype, there is some amount of this, but because the field is there and all, in a global perspective, if you see, it has made tremendous improvement. Same is true for structure prediction and protein design. So we need to take up certain project where these types of protein design or things are this, it need not be entirely structure based. There are, I don't know, some of you, I have noticed a paper on from Microsoft, which was in bioarchive, going from sequence to function without going through structure. So they have trained large language models, which is going to do a similar level of prediction as a structure-based function prediction will do. And they are claiming that, of course, I mean, the example of ESM-fold is that it doesn't use that much of a structure as AlphaFold uses, but this one. So I think what is more important is we need to, I mean, each of us have our own region of interest, but if we want to come up together and expect a funding for training and all this, we should have form a network, and Divarka is one of the similar type of people we could bring in, and we should start a network and take a project with a couple of global overall themes around this, whether it is protein design, antibody design seems to be an interesting area, or could be other types of protein design, or any part of this, some along this couple of bigger questions, and we build expertise. The important point should be more people should come together. We should talk to each other across discipline, and people learn from each other. I mean, some of the things I learned from my lab is my student is working on a machine learning model, or did something random forest or simple deep learning, and found that, oh, my ROC is this. But this paper, which has published used CNN, has done very good result. And I don't get, but on that data set, they have got very good result. Of course, to explain CNN to a biology student who is trained in biologist is not that easy. But if you ask the student, go and look at that data set, why it worked carefully, so better. Okay, you find in those proteins, there are a lot of linear motifs which are governing interaction. So linear motif governing interaction mean only one part of the sequence which is important for governing interaction. And in your machine learning model, you are using the whole sequence. By CNN, you are able to scan this and pick that region or something. So all these things can develop only when we talk to each other and all this. So I think coming up with a group and interaction is going to be the most important part. And next we should think of a couple of common themes on which are common projects, which could be disease oriented, it could be other biomanufacturing oriented, where each of us can contribute. That could be the starting point. And after that, we could take up other projects and all. I think we have to see how the students train. Like as Dinesh said, many of us students who get biologists, they can take certain tool and learn, but they exactly don't learn in the way IIT or a triple IT teaches all these machine learning algorithm or something. But if some bridge can be established, they can have a formal training in machine learning. Same way, a computer science guy could get a quick primer on biology. Somebody like Alok or Anurag or something. You don't have to teach them all biology. You can give only a limited amount of biology, but is important for them, which they can use. And these type of things can only come if you design it in terms of a specific way. And what Dr. Gokhale has been telling in the context of our break is what he calls the fractal courses. The fractal courses means it doesn't have to be a full course. You call an expert on a weekend, he teaches only for 5 or 6 hours on a particular topic, and the students find it adequate to then rest of the things they can search in the net and go forward. So I think that could be the strategy for this area. Thank you.
Okay, any final points from others? If it's not there, I think Debasisa will take forward. Maybe the next activity will plan, and then I will circulate it and get opinion from everyone. I hope all of you will participate in future activities also.
Anurag,
Anurag, you want to say
any-
something?
No, I just turn it on. But since
okay.
I'm speaking, just one.
Go ahead.
Dinesh, I was wondering, did you try the alpha missense that came out in October last year?
Yeah.
So even with AlphaFold, I mean, I'm seeing that it gets better with iteration. So last year using alpha missense, at least on the human side, we were able to start predicting which even small mutations would change function. Now I would imagine that they had far more information to work with on the human protein side compared to the plasmodial, which basically tells you that, they'll get better as more information comes. But that also reminds us a very
But
important.
alpha missense may not be entirely from a structure point of view. They are only predicting whether the mutation is pathogenic or
Oh,
not.
yes, absolutely.
So that is what is. But finally, the goal is to predict pathogenic or not. Why bother whether we understand structure or not? So
I was gonna exactly.
yeah.
So, and on the human side, I mean, to be very honest, that's a very important point. But they've got better. Like in the beginning, they couldn't. Now they.
Yeah, it all depends on what data we have. Lipi also pointed out there is very little information about membrane proteins. So once those information is available with us, maybe these algorithms will get better in future.
And all the more reason that we need.
Unfortunately, parasite proteins they have very unusual genome composition.
Yeah.
And so their amino acid composition also very tricky. And then very few structure has been solved. So the database hardly has.
They might be fundamentally different, Alok. I guess they have completely different evolutionary pressures compared
that's
to
right.
organisms.
So it's a less structural pressure also, a different kind of structure.
Another, I mean, since there is a lot of discussion on the David Baker's work and the type of fantastic protein design work he has been publishing, see, one thing I have noticed over years, the design which David Baker publishes, nobody else is able to do it, even if you use same Rosetta fold or something. And the main reason for that is the selection, the ones which is finally selected for experiment is not entirely by automated criteria. It is driven by intuition. Your design filtered maybe 500 possible design out of 1 million. But out of those 500, if I use David Baker's program, I am start with those 500, and I cannot do experiment on all 500. Neither David Baker does experiment on all 500. The ones which I design probably will work, and this is where his strength is mostly in terms of the ones which he picks for that it is governed to a large extent by intuition. So in any of these projects, when we take it, working on the first hand, you get to know the real power of the method. The same method, you can reproduce it 100%, but you would say that you may not succeed in that. Same true for this recent machine learning based antibiotic design, which was published in Cell and all these. But finally, they tested only 5 in the experiment. Why didn't they test all 500, which they are machine learning this one? So that is where all these methods have certain things which you need to understand better when you actually use it. So that is why, I mean, students should actually be able to replicate each and every work which is published. Even a student takes on one weekend, they replicate entire David Baker's work. They understand much better than they will understand by reading his full paper or things like that.
Completely agree.
I think that's a good idea.
One theme that has emerged from Shachi, Lipi, Dr. Anurag is that we need this back and forth between the computational people and the wet lab people. And they need to, it's almost like you merge, like have a combined lab, and there is like the crosstalk between the two is where the magic happens.
As I mentioned, David Baker has a dozen of people working on just wet labs. So whatever proteins, 50 proteins in a go, they purify and do the folding and look into the structures. So that kind of infrastructural support is the reason they are able to design these proteins.
I think, Dilip, we also need methodology, For example, a quick screen. I mean, like say, if you predict 50, let's say design of 50 protein, I mean, 50 you have done it. Out of that, if one turns out to be all right,
that is where
you have done a good job.
Yeah,
but
that is where selection of proteins is a very important. Say if they pick a PFP.
What I'm saying that checking the activity of 50 proteins.
Right.
You can get them synthesized. You need to have assays which run very fast.
Yes.
So if you want to test confirmation or any of the thing, I think the experimentalists also have to develop rapid method of screening along with that. I think we can probably take it and have a discussion. I think Debasisa will take it forward. All of you, I guess, agree. Anurag will take it forward and organize a one-day workshop.
Completely agree. And maybe we
so
even
it can be jointly done by TSB and Bioinformatics Center.
Yeah, absolutely.
Happy one day workshop where data is presented, computer scientist comes, you people can see our facilities that are there. And And it will be a hardcore data exchange kind of thing. I hope people from outside will be able to come also, and we'll plan it in a way that large number of people can participate in it. And also I'll sort of talk about the kind of training or teaching programs that we are running and we plan to run. I mean, we have a plan to run a teaching program on digital health, and we can incorporate some of these aspects into that also. So to attract computer science students to come to biology. Is that
Great.
okay? So Ashish, can you thank on behalf of the Bioinformatics
Yeah,
Center?
yeah.
Just
Thank if
ladies
Professor
and
Dilip, you wanted to say something?
computational biology. Yeah, I just want to add one more thing.
Dilip.
For computational biology, as we know, cryo-EM is now moving towards the cryo-electron tomography, and the biggest challenge in electron tomography, people are coming with the finding the signal in that whole cellular milieu, like which protein or which complex is where. And that is where I believe, I mean, computational biologists, people, I'll be happy to talk to them. Like we can develop some sort of algorithm to, picking up the signal out of the crowd. And that is where I guess a machine learning can be used. There are multiple groups in Cambridge and other places which are working in this field, and this is going to pick up in future. But there is a huge scope for developing tools in that particular field.
Right. Thank you. So
Okay.
just to kind of summarize and close, thank you everyone. As I mentioned at the beginning, it's much more fun to meet people in person than on a Zoom screen, but constraints with transport have made us do that. But the next one will be in person and hopefully longer duration. And it was just a delight to hear perspectives from everyone, from Dr. Anurag, Dr. Debasisa, and everybody. So just delighted that you chose to spend the afternoon with us. And so couple of key takeaways. One was maybe like a journal club which could meet virtually once a month. And other is maybe so the wet labs and the computational parts are distributed. They're not, it's a little infeasible. Maybe like a distributed consortium which has some computational experts, some protein characterization and validation experts, which could work in a distributed fashion as a consortium. So that is one kind of action item that struck me, but we are happy to brainstorm together and take this forward. And thanks once again for your insights and sharing your time with us.
Thank you, all of you.
Thank you.
Thank you. Thank you very much.
Bye, everyone. Thank you.
Thanks.